All right, let's dive into the incredibly weird and wonderful world of cellular biology. Today, we're going deep on the unusual organelles that let parasites do what they do best, thrive at our expense. And believe me, this isn't just about biological quirks, it's a story of extreme adaptation and, frankly, evolutionary genius.
First, just take a look at this. It's a free-living ciliate, a single-celled organism that's basically a microscopic metropolis. It has everything it needs to hunt, to feed, to reproduce, all on its own. It is complex, beautiful, and completely free. But what happens when a cell like this decides to trade all that freedom for a totally different way of life?
And that really is the big question, isn't it? How do you go from that self-sufficient little beast to a stripped-down, hyper-specialized agent of disease? Well, it's a journey of massive transformation. It's about giving up certain abilities to gain the ultimate prize, a cozy, nutrient-rich home inside another living thing. It's a story of evolutionary trade-offs, of loss, and of some truly radical innovation.
So, to really understand a parasite's toolkit, you've got to understand where it came from. Let's trace the evolutionary path of one of the most important groups of parasites on the entire planet, the apicomplexa.
This diagram here maps out that whole journey. It's wild to think about, but the ancestors of things like Plasmodium and Toxoplasma were probably free-living phototrophs, kind of like the Vitrella you see at the top, basically tiny algae. But then evolution found a new strategy. Some of them became predators, like the culpidillids, which could hunt other cells but also still photosynthesize. That mix-and-match lifestyle was the critical stepping stone to becoming the full-blown parasites we know today.
And this gets at a really fundamental idea in evolution: cost versus benefit. I mean, why would you bother keeping all the complex, energy-hungry machinery for photosynthesis if you can just steal all your food from a host? The shift to parasitism meant shedding those now useless functions and building a brand new, sophisticated toolkit for one job and one job only: invading and surviving inside another organism.
So what are these specialized tools? Let's have a look at the machinery a parasite uses to, well, break and enter.
Meet the apical complex. You can think of this thing as the parasite's molecular Swiss Army knife. It's this highly sophisticated set of structures right at the pointy end of the cell, including secretory organelles called rhoptries and micronemes. Its whole purpose is to orchestrate the invasion, squirting out this cocktail of proteins that messes with the host cell and paves the way for the parasite to get inside.
Right under the parasite's skin, you find this incredible structure, the inner membrane complex, or IMC. On the left, you've got the schematic, this system of flattened vesicles, all reinforced by a protein mesh and microtubules. But then look to the right, that's the electron micrograph, the real deal. You can actually see those layers. This whole setup gives the parasite the structural support it needs to physically force its way into a host cell. Plus, it doubles as a scaffold for building new daughter cells. Super efficient.
Now, parasites don't just invent brand new machinery. They are masters of repurposing and downsizing old parts. And that brings us to the very strange case of the parasitic mitochondrion.
This table just lays out the spectrum of this reductive evolution perfectly. In a parasite like Toxoplasma, you still find a pretty recognizable mitochondrion with an electron transport chain. But in an anaerobic parasite like Trichomonas, it's been reduced to a hydrogenous zone just for anaerobic metabolism. And then you get to Giardia, and it's stripped down even further to a mitosome, whose only known essential job is building iron-sulfur clusters. It's a clear path of simplification.
So you've got to ask, right? If the main powerhouse function is gone, why on earth would you keep the organelle? Well, the fact that they all hang on to iron-sulfur clustered biogenesis is the big clue. This process is absolutely vital, but it can also be pretty toxic, creating reactive oxygen species. So the leading theory is that keeping the organelle, even as a tiny remnant, is all about compartmentalizing that dangerous but essential chemistry.
And speaking of mitochondrial leftovers, one group of parasites has taken its mitochondrial DNA and organized it into one of the most bizarre and complex structures in all of biology. Period.
Check these guys out. These are trypanosomes, the parasites that cause sleeping sickness. And do you see that bright blue structure inside each one? That's called the kinetoplast. It's a single, massive, interconnected network of mitochondrial DNA. There's nothing like it in our cells.
Okay, so this diagram shows you just how intricate this whole thing is. The kinetoplast DNA, or kDNA, isn't just a simple loop. No way. It's a massive network that's physically tethered to the base of the flagellum.
That physical link is critical. It makes sure this incredibly complex ball of DNA gets sorted out correctly when the cell divides. The network itself is made of two different types of DNA circles, all linked together.
Honestly, the best way to describe this structure comes right from the source material. Thousands of these DNA circles are catenated to form chainmail. Just picture that. They are interlocked like the rings in medieval armor. This creates a structure that's both flexible and incredibly strong, a single gigantic linked molecule of mitochondrial DNA. It is absolutely mind-bending.
So what's in this DNA chainmail? Well, you've got about 25 of these big maxi circles. They're what you might think of as normal mitochondrial DNA, encoding key proteins. But the vast majority of the network, we're talking like 90%, is made of thousands of tiny little mini circles. And their job is just bizarre. They encode things called guide RNAs, which act as templates for a massive process of RNA editing. They literally go in and add or delete bases to make the messenger RNAs from the maxi circles functional. It is an astonishingly complicated system.
All right, so beyond invasion and wild genetics, parasites have also evolved some highly specialized tools for very specific jobs, often tied to a particular spot in the host or a certain stage in their life cycle. Take Giardia. To cause the nasty diarrhea it's famous for, it has to physically cling to the wall of your intestine, and it does this with its ventral disc, a huge, complex spiral of microtubules. This thing basically generates suction, acting like a powerful little suction cup that lets the parasite hang on for dear life against all the forces trying to flush it out. No disc, no disease. It's that simple.
And now for Plasmodium, the malaria parasite. Its life cycle, which you can see on the left, is ridiculously complex. But if we zoom in on the Eukaryotes stage, that's the motile form inside the mosquito's gut, we find something really unique. The images on the right show crystalloids. These are dense, membraneless organelles just packed with proteins. We're still figuring out exactly what they do, but we know one thing for sure. They are absolutely essential for the parasite to develop into the next infectious stage, the sporozoids. They are like a temporary, purpose-built factory that's critical for transmission.
So when you look at all of these disparate, frankly bizarre structures, what's the unifying theme? What ties them all together? Well, the answer is in the messy but brilliant process of evolution itself.
See, evolution doesn't design things from scratch like an engineer. It does what the biologist Francois Jacob famously called evolutionary tinkering. It takes parts that are already there, and it repurposes them, it reduces them, and it smashes them together in new ways to solve a problem. These strange organelles are the perfect product of that practical, jury-rigged process.
And you can see it everywhere we've looked today. The apical complex, that was likely adapted from a structure that a free-living ancestor used for predation. The apicoplast, that relic of a photosynthetic past, it ditched all the genes for photosynthesis but was kept around for other essential pathways. The big takeaway here is that evolution isn't looking for the perfect, most elegant solution. It's a tinkerer, just trying to find a solution that's good enough to work.
And this leaves us with a really fascinating final thought. The parasitic lifestyle is the ultimate in minimalism. If something isn't absolutely essential, it gets thrown out. So that means these bizarre, complex structures that are left behind have to be fundamentally important. For every organelle we understand, like the ventral disc, there are mysteries like the crystalloids. The study of these weird organelles is a real frontier, reminding us just how much we still have to discover. What other vital functions are still hidden inside these evolutionary marvels? Answering that question is one of the great, exciting challenges in parasitology today.